In this video, we are going to look about an important topic which is sudden blotting. So, what is this sudden blotting? The detection of specific DNA sequence using blotting and hybridization technique is sudden blotting. So, DNA is detected. How it is done? There are my five important steps. So, first is DNA digestion. So, DNA is cut into small pieces, and second is DNA separation using agarose gel electrophoresis. Third is blotting. Fourth is DNA hybridization. So, the process of joining the probe DNA, which contains our specific sequence complementary to the target DNA, and last is DNA detection. In this picture itself, you can see the overview. So in the gel, there is a DNA or RNA sequence. It is separated using electrophoresis technique, and then blotting has been done. So the DNA samples are transferred to a membrane using blotting technique, and then the membrane is taken, and there's small red color lines are there. So the probes are attached. So the probe will attach to our target DNA, and it is visualized using deduction techniques like autoradiography or chemiluminescence. So this is the overview. So let's check the intricate details of this technique. So first step is DNA digestion. So we can't be analyzing a DNA like as it is. It might be very long, so we can't be separating it or analyzing it. So we have to digest it. That means we have to cut into small pieces using restriction enzymes. So for example, if we take DNA from E. coli, we extract DNA, genomic DNA, and then use restriction enzymes such as ECO type 1. So this will cut at a specific sequence and produce small fragments of DNA. So this is DNA digestion. Second step is DNA separation. So the DNA is separated based on their size using agarose gel electrophoresis. Already I have made a detailed video of agarose gel electrophoresis. You can check it out. Anyway, I will just share the glimpse of it. So as the DNA are negatively charged, it will start moving towards positive electrode. So the smaller DNA molecules will move faster, and it will get settled in the bottom easily. But the larger DNA molecules will move slower. Thus, the gel will separate DNA based on their size. Third is the most important step of this part. So blotting. So blotting technique, what we use is we just take a tank and we will fill a buffer in that. So alkaline buffer is filled, and we keep a sponge. So you can see in this picture that white color big chunk is the sponge, and we keep the gel over the sponge. And over the gel, we keep the nitrocellulose membrane to which we transfer our DNA. Over the membrane we keep a paper towel, and over the paper towel we keep a weight. So what will happen if we keep a weight on the sponge will absorb the water and it will get shrunk. So while absorbing the water, it will transfer the water, and due to capillary action, the DNA in the gel, it will get printed into the nitrocellulose membrane. So just like taking a print, so the weight on the top apply pressure on the setup, and the buffer solution moves upward towards the sponge, through the sponge by capillary actions. So capillary action is an important term. The buffer processes the membrane and reaches the paper towel. The DNA present in the gel is imprinted into the nitrocellulose membrane. So this is we call as blotting. Fourth is the important step, which is DNA hybridization. So the double-stranded DNA is denatured due to denaturating buffer. So if DNA is in double stranded, we can't be binding our probe DNA. So it has to be denatured, and it has to become a single stranded so that our probe DNA can be bound. So the double-stranded DNA is converted into single-stranded DNA, and we add blocking agents. So which we use is dry milk. We add blocking agent because our probe might fix in some other places also. So if we put blocking agents, means our probe will attach it only to the specific target DNA. Okay, so the probe is hybridized with the target DNA sequence, and the probe is radio labeled with P32. So when we look in visualization technique, it will glow, and we can identify whether it is attached and our target DNA is present in the sample or not. So unbound probes are washed. So and another thing, the how we prepare the probe, because if we need a target DNA, we prepare the probe in a complementary sequence. If for example our target DNA sequence is AATTGGCC, means our probe should be in TTAACCGG. So the complementary sequence should be there so that it will bind to the DNA, and we can identify whether our target DNA is present in the sample or not. So the last final step is DNA detection. So we detect whether our sample contains the target DNA with a technique called autoradiography. As I said, we attach the radio label with a probe, which is P32. We apply some radiation, and at that radiation, our radio labels will glow. So at that time, we can realize, okay, there is our sample contains the target DNA. If there is no glow, means the target DNA is not present, or the probe didn't attach to the sample target DNA. There are other technique also like chemiluminescence, in which instead of the radio label, an enzyme will be attached. So when we give the appropriate substrate, that substrate and enzyme will react, and they will produce some fluorescence or luminescence. With that also we can identify whether our target DNA is present or not. Coming to the applications, there are many applications for this southern blotting. So most important applications are gene discovery, whether a gene is present or not. We can identify the specific genes, and genetic mapping. So like how DNA sequences are arranged, these kind of research can be done. And diagnostic tools. Some genetic disorders will have a specific genetic marker. So with that sample, we can identify whether that genetic marker is there, so that that disease is there or not. We can diagnose this southern blotting. It's also used in forensic studies and DNA fingerprinting. So coming to the northern blotting, here we detect the RNA. Most of the steps are same like southern blotting, but there are small differences. So what are the differences are? First, there is same southern blotting technique only, but we use formaldehyde treatment to convert the secondary structure of the RNA into linear structure. So how we use a denaturing agent there, here we use formaldehyde treatment. So to convert the secondary structure to linear structure. Second, then same agarose gel electrophoresis, same blotting technique. Here there is a small difference. We use aminobenzyl oxymethyl paper. Oh wow, what a great! So there we use nitrocellulose membrane, but here we use aminobenzyl oxymethyl paper because it has more affinity towards RNA. So thus we use this paper, and the same blocking procedure. And there is no denaturing agent because already RNA has single stranded, so we no need to use any denaturing agent. And same hybridization technique and the same visualization technique like autoradiography or something. So application for this northern blotting is to identify and study expression of genes, whether some genes are expressed. So if it is expressed, means RNA will be produced. So for the detection of gene expression, we use this northern blotting. So thus in this video we just covered the principle and how the procedure and applications of northern and southern blotting. So that's all.